started. So I'm Christopher Schmidt. I'm an assistant professor on the Charles River campus in anthropology and biology. And today it's my great pleasure to welcome Dr. Benn Torres to the Genome Sciences Institute seminar series. Dr. Benn Torres, he uses she, her pronouns, is the director of the Laboratory of Genetic Anthropology and Biocultural Studies at Vanderbilt University in Nashville, Tennessee, where she's associate professor of anthropology. Dr. Benn Torres earned her BA in pre-professional studies, anthropology and computer applications at the University of Notre Dame, her master's and PhD in anthropology at the University of New Mexico in Albuquerque, where she worked with Dr. Keith Hunley and Dr. Ann Stone, and she completed postdoctoral training in genetic medicine with Dr. Rick Kittles at the University of Chicago before being assistant professor of anthropology at Dartmouth prior to moving to Vanderbilt. Her primary research area, as we'll hear today, is the Anglophone Caribbean, where she explores genetic ancestry and population history of African and indigenous Caribbean peoples. And a second emerging area of her research combines the tools and theories of genetic epidemiology with anthropology in order to holistically examine health disparities, in particular focusing on women's health, with a specific focus on uterine fibroids among women of African descent. Her book, Genetic Ancestry, Our Stories, Our Past, co-authored with cultural anthropologist Gabriel Torres Colon, was published by Routledge in 2020. And her work has been recognized with the highest accolades in her field. She's president of the American Association of Anthropological Genetics, the recipient of the Gabriel Ward Lasker Award in 2017 for the most significant contribution to the journal Human Biology, and of the 2021 Robert W. Sussman Award for Scientific Contributions to Anthropology by the American Association for the Advancement of Science. Today she'll be talking to us about genomic ancestry and cultural identities, case studies of community-engaged genomics in the Caribbean. Please join me in welcoming Dr. Benn Torres.
So first off, I'd like to thank you all for the invitation to come here and share some of my work, and for Chris for actually sending the invitation, and Abby making everything just sort of work beautifully. I'm really excited to be here and just share a bit of some of the work I've been working actually for a little while in the Caribbean. In today's talk, I really just want to focus on these aspects of community-engaged genomics. Particularly when we think about genomics, it's often people that are either in the lab or behind their computers. But what happens when you leave the lab and start talking and interacting with people? So I want to focus a bit on that and the implications of that, not only for doing our own science, but thinking about how the science is done and building relationships between people who do the studying and those who are studied. So as a genetic anthropologist, I've been interested in using genetics and now genomics to address questions about how the dynamics of human experience shapes patterns of human genetic diversity. Through my work in the Anglophone and now Hispanic Caribbean, I've explored genetic ancestry and population history of African and indigenous Caribbean peoples in an effort to eliminate histories and perspectives that have been systematically erased or otherwise marginalized. Central to my efforts to address these questions is the idea that human genetic diversity reflects the intertwining nature of biology and experience, and that by studying these interactions, these sorts of relationships, we can learn more about what it means to be human. So in my work, I tend to rely upon community-based participatory research, or CBPR, and this is a framework that's been developed in public health. And it can be defined as a research approach, not a methodology, but just a framework or an approach that equitably involves community stakeholders and researchers in every aspect of the project. With this approach, all partners contribute expertise and shared decision-making and ownership. And this particular approach actually has implications for how studies are designed, how the results might be disseminated, and then how that data might be archived or steward or maintained. So with this approach, community-based participatory research and other related terms, so I think center is another one, community-engaged research is another term. As I mentioned, these were developed in public health, but it's gaining momentum outside of public health. So what I have shown here is a screenshot from a 2015 article that was published in The Atlantic. And in this article, the author, Ed Yong, he talks about community-based approaches being used to learn about health disparities as they impact minoritized communities. So to quote the author, he says, rather than the usual approach in which researchers helicopter in, do their thing, and tell participants what they found under the rubric of engagement. CBPR requires communities to actively and equally participate in all phases of the research, from planning to publication. They become genuine scientific partners, not guinea pigs. In addition to using community-based approaches, in my research I also use biocultural perspectives, meaning that I purposefully situate my central study questions within both biological and cultural contexts. I tend to focus on how the ways in which people identify and organize themselves, how that impacts patterns of genetic variation. And it's through this work that I can reimagine and reclaim knowledge about the experiences of African and indigenous peoples throughout the Caribbean. So to date, much of my focus has been using genetic ancestry testing to examine signatures of population migration and interaction. A variety of scholars have rightfully argued that the uncritical uses of genetic ancestry testing can be quite damaging in that it can reify biological notions of race. And this is an important point, one where researchers and actually the lay public consistently make. And it has reverberations not only in fields like anthropology and other social sciences, but also in medicine, right? So how people understand this relationship between genetics and social identities and constructions of race. Despite the problems, or the potential problems with genetic ancestry, I also regard these technologies as holding potential to preserve and reconcile histories of marginalized communities. I'll give you some case studies that will show you how I do this or how I approach this. As I think about some of the various projects that I've completed across different Caribbean island nations, I've also begun to reflect on how changing technologies and ideas about ethical research impact how researchers engage with communities of participants, as well as our own scholarly communities. Today I want to share some of my own experiences of doing community-engaged genomics, really to illustrate the utility of engaging with broader sociocultural contexts in which genomic data emerge. So to do this, I'm going to highlight three projects, or I guess two projects that I have been involved in. I've got several across the islands, but I'm going to focus on a project that I did in St. Vincent and in Trinidad, and then my newest project in Puerto Rico. So here I have a map showing the locations of some of the communities I've worked with. In particular, the Garifuna in St. Vincent and the First Peoples community of Santa Rosa and Arima in Trinidad. These are indigenous communities, or self-identifying indigenous communities in the Caribbean. My work in Puerto Rico, I work with self-identified African descendants in Puerto Rico. And as we get to, as I talk a little bit more about Puerto Rico, I will emphasize the importance of self-identification, particularly in the context of Puerto Rico. So in all of these projects, I've used genetic and now genomic data to add novel perspectives to understanding how communities came to be how they are. Because of some of the history in the region that's been lost to time, or even as a result of systemic marginalization, in which both African descendant and indigenous Caribbean populations were written out of history, genetic data offers some potential to redress some of the gaps in knowledge about these communities. So in the first case study that I'll talk about, this is some work that I did with colleagues at the University of Pennsylvania, at Vanderbilt, at National Geographic, as well as community partners in both St. Vincent and Trinidad. This work has been published, these papers came out in 2015 and in 2019. So I'm only going to talk about small aspects of each of these studies. So in those projects, one of the questions that we addressed was about the nature of African and indigenous interaction within the Caribbean. So beyond cultural knowledge exchange, and this is already evidenced in ethnohistoric sites by archaeology, one thing I wanted to know was how extensive was gene flow, okay? So genetic exchange between African descendants and indigenous Caribbean peoples. And this is a question that both communities were interested in, because much of the existing knowledge about colonial era Caribbean comes from what European observers noted. And these observers wouldn't have been privy to the internal workings and lives of indigenous and African descendant communities. Consequently, contemporary communities actually want a better idea and they want to know what genetics might offer to their understanding of the past. So pictured here are actually buttons from the late 18th century, and on them you can see these images of indigenous Caribbean folks and kind of African descendants in the Caribbean, and you see these interactions, and this kind of served as a point of me kind of thinking about and being motivated to address these questions. So prior to doing any of the studies, the project was reviewed and approved by our IRBs at our respective universities, so University of Pennsylvania, the University of Notre Dame, and then when I moved universities at Vanderbilt as well. In addition, as part of doing this work, we also obtained ethics reviews and approvals from the Ministries of Health in St. Vincent in Trinidad, as well as approvals in the form of advocacy from local community organizations. Right? In addition, as part of the study, each study participant provided written informed consent prior to sample collection and any documentation of family history or genealogical information. So to give a little bit of background, with St. Vincent, we ended up collecting a total of 64 samples from Garifuna or Kalinago peoples. So the history of indigenous communities in Puerto Rico is actually a chronicle of resistance and survival. In collaboration with African peoples, the indigenous peoples of St. Vincent actually waged two major wars against the British in the 18th century. They won the first one, and in the second one they didn't win. Okay? And what ended up happening is that many people were exiled, actually first to an island known as Balaku. Okay? It's basically a rock in the ocean. And kind of were forced to live there for months at a time. Large numbers of people just died of starvation. There's like really no vegetation on it. And then in the end, people from that sort of island were then exiled to Central America. So to Roatan, Honduras, and then the community has actually since spread across Central America, and there's actually a Garifuna diaspora in New York. After this exile, not everybody was exiled, and then some people actually came back. So there is an indigenous component or contingent on this island. So the indigenous communities that my collaborators and I worked with were located primarily in the north of the island, in Oya, in Sandy Bay, Fancy, as well as some areas in the south and southwest regions of the island. So Greeks Point, Rosebank, Rose Hall, and in the capital, Kingstown. So we were interacting with folks and collecting samples and getting really to know about the history of the community, as well as contemporary ideas of what it means to be Garifuna. We also worked in Trinidad. So in the north central region of Trinidad, roughly 26 kilometers east of the capital of Port of Spain, is an area called, or a town called Arima. And Arima is home to the Santa Rosa First Peoples community. And the history of the First Peoples community in Trinidad actually extends back to when Trinidad was a Spanish colony at the end of the 16th century. In an effort to annex indigenous lands, the Spanish government actually consolidated the island's native peoples into missions. And the Santa Rosa mission was the final residence place of many of the remaining indigenous peoples. And a concentration of indigenous Caribbean people actually remained there, though native peoples can be found scattered in the north regions throughout the island. So today, many of the people that self-identify as indigenous can be found in Arima and a nearby town called Lopino. Interestingly, many of the people in these communities who identify as Spanish, not as Spanish, but as indigenous, they actually have Spanish surnames. Okay, so this is a marker of indigeneity, having kind of a Spanish surname. Some of the elders actually still speak Spanish, and they're known for a particular type of music. This music is played more often during Christmas time, and these songs are usually sung in Spanish, and they're played with a 4-string instrument called a cuatro. Okay? And the musical style is parang, is what it's called in Trinidad. In Spanish-speaking islands, you'll see similar types of music known as parangas. Okay? So you have these kind of connections across the islands that kind of extend back in time. So in Arima and in Lopino, we were able to collect data from 36 people in these communities. So you'll notice with both the Garifuna in St. Vincent and the First Peoples community in Trinidad that my sample sizes are really, really small. Okay? And this is really a reflection of the small size of these communities. In Trinidad, the indigenous population makes up only 0.11% of the entire population. Okay? In St. Vincent, self-identified indigenous people represent only 3.6 of the total population. So these are not huge communities in the first place. Sampling was also affected by shared relatedness, as well as those who ultimately agreed to participate in the study. Okay. So in this project, just to kind of clarify, one of the goals was really to look at peopling of the Caribbean and to understand more about the biological relationships between indigenous people in the islands and then indigenous peoples in the circum-Caribbean area. So in terms of our workflow, we looked at uniparental markers, so mitochondrial DNA and Y chromosome DNA, to say something about maternal ancestry and paternal ancestry, respectively, and we also looked at autosomal DNA. So looking at those maternal lineages, we focused on mitochondrial DNA, and specifically, we sequenced a portion of the mitochondrial DNA, so hypervariable sequence 1 and 2, okay, so we actually had sequence data, and then we diagnosed or identified mitochondrial haplogroups via SNP genotyping. Okay. With this data, we then did kind of your standard population genetic analyses and phylogenetic work. For the Y chromosome, again, this really focused on paternal ancestry to say something about the male lineages. We genotyped Y chromosome short tandem repeats, so these repeating units on the Y chromosome, as well as identifying Y chromosome haplogroups via SNP genotyping. And then again, did comparable summary statistics using population genetics and phylogenetics. We also looked at autosomal ancestry. Okay? And for autosomal ancestry, we used a version of a microarray known as the Genochip 2.0. So at the time of doing this study, National Geographic actually was coming to the end of their genographic project, and this was a global effort to use DNA to learn more about how humans have migrated and populated across the globe. I was part of that in terms of focusing on peopling of the Caribbean. So there was a specific array that was developed for this work. In this version of the array, Genotype 2.0, it contained 135,000 autosomal short nucleotide polymorphisms, or SNPs. Okay, and then you can see the breakdown of these SNPs on the Y chromosome, and then SNPs on the mitochondrial DNA. The basic analytical workflow is kind of standard. You do your quality control measures, which then cleans up your data set, but then you typically have a reduced number of SNPs that you look at. In this case, we lost a few people through quality control because they were either related or the DNA genotyping didn't hit a particular threshold. In terms of our data analysis, we merged our data with 1,000 Genomes and Human Genome Diversity Project reference populations to kind of get an idea of comparisons across the globe, and then specifically focusing on the circum-Caribbean area. Okay? And then we looked at principal components and estimated global ancestry. So I'm going to go through this pretty quick because I do want to get to the kind of community engagement part. So here I have a histogram showing the distribution of mitochondrial haplogroups. So this is mitochondrial DNA. It's informative about maternal ancestry. And what's really interesting about mitochondrial DNA is that it is inherited from a mother to all of her children. So everybody in this room has mitochondrial DNA, and it looks like your mom's. Okay? And hers looks like her mom's, hers looks like her mom's. So it goes along that maternal line, and we can look at it back in time. And based on phylogenetic similarities, we know that for the most part, and there are, of course, exceptions, that these mitochondrial families or haplogroups, they're continent specific, right? So we can figure out what your haplogroup is, we'll know where it's common in the world, and we'll say that you have ancestry likely from X place in the world. So looking at these haplogroups, I have it kind of broken down here by continental ancestry. And the two communities that I've underlined here are those from St. Vincent de Garifuna and then the First Peoples community in Trinidad. And there's a couple of striking points that you can note here. First off, with the First Peoples community in Trinidad, they have a very high number, high frequency of indigenous maternal ancestry. And this is one of the first studies to show this in Trinidad. So these communities that have, for generations, said that they are indigenous, there is genetic data to support those claims. Not that it was necessary, but it is part of that. In addition, you could see that the Garifuna and St. Vincent, they also have a fair amount of indigenous ancestry, but they also have a fair amount of African ancestry, which aligns quite nicely with their own oral histories of how these communities came to be. Okay? You'll also notice some similarities across the Spanish-speaking islands, so Puerto Rico, Cuba, and to a much less extent Dominican Republic. But you also see high frequencies of indigenous maternal ancestry. And a lot of this really has to do with the ways in which the Spanish colonized islands versus the ways in which the British and French colonized islands. And we see this reflected in patterns of genetic ancestry. Right? There's actually a lot more that I could say about this, particularly about the Accompong Town Maroons, which are an Afro-Indigenous group in Jamaica. Right? But we're going to focus on the Garifuna and the First Peoples community. So if we look at the Y chromosome, so focusing on the paternal ancestry, you'll see that both Trinidad and St. Vincent have paternal ancestries from Africa, the Americas, and Europe. Now what's particularly interesting in the case of both Trinidad and St. Vincent is the discovery of this haplogroup QM3. Okay, so it's depicted in red, so right there. Haplogroup QM3 is what we commonly see in the Americas. Okay, so this is telling us that along the paternal lines, for many of these people in these communities, it directly traces to indigenous peoples, okay, on the paternal lines. And what's interesting about this is oftentimes many people in the Caribbean and beyond are taught that indigenous people were all killed shortly after the arrival of the Spanish, that they did not live. Okay? And what this data is suggesting is that not only did they live, but we can see elements of their ancestry in people today. And there are people who identify as indigenous. So again, we see that biological connections between the past and the present, and some that directly contradict some of the stories that are told about these communities. If we look at this very busy slide, this is an image of one way we can depict global ancestry for both St. Vincent and Trinidad. Okay? So if we look at kind of this image first, this is kind of a structure plot. You look at these basically in columns, and the different sort of color blocks represent different ancestries. So everything kind of listed here are reference populations, so 1,000 genomes populations and human genome diversity populations. Okay? For points of reference, this is Trinidad and St. Vincent, and then one reference group from 1,000 genomes, the ACB. Those are groups from Barbados, okay, so another Caribbean community. Okay? And then this slide here, this part of the slide on the bottom, this is basically these last two columns sort of blown up so you can see things. Basically what these structure plots are showing is that in St. Vincent and Trinidad, there's primarily kind of three major reference groups. So we have African ancestry, Native American or Indigenous Caribbean ancestry, and European ancestry. For these particular analyses, we can get into a little more finer detail and also look at East Asian and South Asian ancestry. South Asian ancestry was particularly important in Trinidad because shortly after emancipation, indentured laborers from India came into the island and actually make up half of the population now. So we see kind of that history also reflected in this indigenous community. Another way of sort of interpreting these structure plots are actually to look at numbers. So here you can see that St. Vincent and Trinidad have roughly 10 and 12% indigenous American genetic contributions to their genomes respectively. In St. Vincent, we see about 70% African ancestry. In Trinidad, it's about 30%. And then the numbers vary a little bit more for European ancestry. Right? One interesting thing that I noted here was that between St. Vincent and Trinidad, the populations are somewhat genetically distinct from each other, meaning that their patterns of ancestry, they look a little bit different. And this really just reflects different histories in the islands. This same idea is reiterated in our principal components plot. So you can read this PC plot in a similar way you might read a map. If points are close together, it means they're genetically similar. If points are further apart, a little more genetically distant. To give you points of reference, this area here, kind of with these blue colors, etc., these are different African reference populations. So you just have different reference groups from Africa. In red, you have different European groups, and then you have groups from the Americas kind of represented in this part of the plot in East Asia down here. So Trinidad is represented kind of by these brown plus signs, and then St. Vincent de Garifuna are represented by these green triangles. And there's a couple of really interesting elements that pop out in this PCA plot. First off, is that the Trinidadians, the people from Lopino and Rima, they're all pretty genetically similar. Okay? And this is after quality control measures, so we know that these folks are not close relatives. Okay? So this can tell us either that they're all genetically similar, or that the reference groups that we have here, they're not accurately capturing the sort of variation or where these folks are coming from. Okay? You'll also notice that for the Garifuna, they're really smeared across this plot, okay, indicating a lot of variation. People who identify as Garifuna, there's a lot of variation in their own ancestry. Right? So, as I interpret this plot, it's really suggestive that the history of African experience is quite heterogeneous across the islands. Okay? So this is an area that we could probably explore in a lot more detail, and again, having that historical and contemporary context of why we see these patterns is very important for making sense of the genetic data. Another thing we looked at was sex-biased gene flow, so understanding whether it's females or males making genetic contributions to the contemporary community. And what we can see overall based on these plots is that there's higher proportions of ancestry from African and indigenous Americans relative to European ancestry along the X chromosome. So this is telling us that there is some evidence of sex-biased gene flow. Within the sample group from Trinidad, we see higher female ancestry contributions from Africans and Americans, indigenous Americans, and then lower female ancestry contributions from Europe. And again, this looks a little bit different than what we see in St. Vincent, also suggesting differences in demographic histories, and this is reflected in the DNA. So what this work really showed is that indigenous Caribbean and African peoples constructed communities together that went beyond simply the exchange of cultural information. Okay? They got together, they exchanged knowledge, and they had babies. And these communities persist in the Caribbean today. Furthermore, we noted that the extent of admixture actually really varied in relation to community history and geography. And when we think about the Caribbean sort of overall, we see that these interactions appear to have left more noticeable signatures in places that served as refuges for indigenous Caribbean peoples. So places like St. Vincent, places like islands like Dominica, as well as Trinidad. So with regard to the community engagement, we first became aware of the social and political context of these communities as actually unrecognized indigenous peoples. Throughout the Caribbean, there's a misconception that all indigenous people were driven to extinction with the arrival and expansion of European colonization. Other people subscribe to the idea that due to generations of admixing, no one can be considered, or at least properly considered, indigenous to the Caribbean anymore. Okay? And the communities that we work with, as I mentioned, they self-identify as indigenous communities. They have actively been fighting these sorts of misconceptions for generations. Related to these sorts of misconceptions was also this idea that being indigenous was somehow socially unacceptable or undesirable. And this sentiment was illustrated by the quote I'm showing here, which was part of a conversation I had with a Garifuna woman. So over the course of the field work in St. Vincent, she and I became well acquainted due to her interest in the study and her knowledge of the community's history. In the conversation that I'm quoting here, she mentions, she's actually just commenting on the ease with which I was able to recruit for the project at local community college. She said, suddenly everyone is Carib. These are old-fashioned words, but these are the words that people use. She said, before, it was an insult to say that someone was Carib. It meant that you were stupid or loose, meaning sexually promiscuous. But seeing you studying this and talking about our history, it makes people less ashamed to be Carib. And I share that little vignette with you to make the point that community engagement was part and parcel of this project. It actually began well before we went to St. Vincent or recruited for the project. During the development of the study, I actually spent a lot of time learning about the history of the peoples of St. Vincent and Trinidad, and from this we were able to develop a central question that resonated with the political and personal agendas of community members in these island nations. Because of histories of marginalization and exploitation, it was crucial to have community buy-in and investment in the study. Furthermore, we decided to partner with community leaders in the implementation, dissemination, and the archiving of the project. 3 of our community partners are actually co-authors on both of the papers, and they retain ownership of the generated data. Okay? What this means is it limits how the data can be shared. Requests for data sharing actually have to go through our community partners, and without their express consent, the data cannot be shared. Okay? And again, this has impacts on how we conduct science and how we engage with one another as a scientific community. Now, we did this, we kind of thought very explicitly about this issue of data sharing. We did this as a way really to acknowledge and respect the autonomy of the community. The second project that I want to share with you to kind of illustrate the complexities of community-engaged genomic work is an ongoing project in which I've partnered with a cultural anthropologist, so Dr. Gabriel Torres and sociologist, and my local community partner, Maricruz Rivera. So looking to previous population genetic studies of contemporary Puerto Rican populations, we can note a history of movement, okay? And with this movement, we see genetic contributions from indigenous Caribbean peoples, from African peoples, and European peoples to the general populace of the island. So in the early 2000s, there were a series of studies, genetic studies, that confirmed the island's tricultural heritage from Spain, from the Americas, and from Africa. However, one of the most cited aspects of the body of work was the high percentage of maternal indigenous ancestry. So upwards of 60% of the sample population belonged to an indigenous American haplogroup. So that's kind of what I was showing you a few slides back where you saw that on the histogram, that tall orange bar. So with that work, through doing preliminary field work, it was back in 2017, community members in this particular area in Puerto Rico, they expressed their dissatisfaction with the manner and the way in which those studies were conducted, and how the results were presented and then received by other Puerto Ricans. These earlier studies tended to use a census-based sampling scheme, and while in many ways it can be appropriate to use a census sample or census-based sample within a population, it's also vulnerable to the same problems that complicate accurate census counts. Specifically, for a variety of historical and political reasons, marginalized populations tend to be overlooked or undercounted in censuses. Okay? So sampling in that way for a genetic study will replicate similar problems. Okay? In this case, many of people who identified as Afro-Puerto Ricans, they were actually not well represented in either censuses or in genetic studies that used census sampling. So as a result of that, many of the community members that I encountered perceived some of those earlier studies as not accurately representing their communities, and they felt excluded from island-wide pictures on the genetics of the island. In addition, the emphasis on indigenous American ancestry tended to occur with the exclusion of other, but namely African ancestry. So in our project, we really aimed to address these issues by explicitly focusing on marginalized populations and bringing together genetic ancestry with culturally constructed ideas of blackness in Puerto Rico. So for the genetic component of this work, I have listed here some of the central questions that we and the community are particularly interested in. Okay? Much of this work really emanates from a community center that I'm working in. It's Corporación Piñones Se Integra, or COPI, and this is located in the Piñones sector of Loíza in Puerto Rico. I'll show you on the next slide where this is in Puerto Rico. So the main goals of that project really were to focus on the genetic origins of Afro-Puerto Ricans with specific attention to African origins, admixture, as well as local and regional variation. In terms of the non-genetic components of the study, we're also interested in learning how Afro-Puerto Ricans understand biological and cultural dimensions of their past. How do they make linkages between the past and the present? And then what happens when you give them this data, genetic or genomic data? How do they think about that? The third thing we want to do is to really facilitate local artistic interpretations of genetic ancestry and community history. So the idea, again, is to return these genetic data and then to see what this community does with the data and how they understand it and how it shows up in their own sort of medium. So here's a map showing some of the sample areas where we've collected samples. So this project has been a labor of love. I began it in 2017 and then it had to pause because of Hurricane Maria. We started up again in 2019 and then a global pandemic hit, right? So it stopped and started and stopped and started, and it has been difficult to maintain contact with participants. But again, there's community buy-in, so they do, people periodically do check in. So in terms of addressing the sampling issue, we actually employed several qualitative and ethnographic methodologies in order to obtain a more critical and attentive approach to population as it exists and as it's experienced among Afro-Puerto Ricans. Okay? We spent a large amount of time, over 5 months, over the course of 2.5 years, really just hanging out and being participant observers. In addition, my collaborators have conducted structured and unstructured interviews where they collect information on genealogies, on family and community histories. We talk also about racial classifications and their understanding of genetics and genetic ancestry. We also talk about the spatial distribution of Afro-Puerto Ricans, both past and present, as this can again be informative of movement across the island. So here, what's marked on the map are some of the main areas where I've collected some samples. And again, the project is growing. I'm hoping to go back actually next month to complete sample collection and work with the communities. So the basic workflow for this particular project is we get the DNA, we do our own extractions, and then we do whole mitochondrial sequencing. We also use pyrosequencing to determine Y chromosome haplogroups, and then we also use a microarray. For a subset of 30 people, I used National Geographic's last microarray, so it's GenoChip 2.0 next generation. And that microarray has 750,000 genome-wide SNPs. And then we look at, we use that data to look at a variety of markers across the genome to focus on uniparental markers, so mito and Y, and then we do ancestry using the methods listed here. With our quality control for our first sort of subset, we still have good data for 30 people, and instead of having data for 750,000 markers, we have it for 670,000 markers. Okay. So what our preliminary results are showing, and this is mitochondrial DNA, we have some of the same populations that you saw, comparative populations that I showed you kind of in the previous slides. They're represented here. But here on the end, these are the Afro-Puerto Ricans, right? So the current study. And what you can see is that the distribution of these haplogroups looks a little bit different than general populace for some of those earlier studies. The little stars that I have listed here, these indicate statistically significant differences in the distributions of the different proportions of haplogroups. Okay? So we see that between general populace Puerto Ricans and Afro-Puerto Ricans, there's a statistically significant difference in these mitochondrial haplogroups, right? So there's different ancestries, there's different stories here. Those sentiments about not being represented in those general studies, that's actually supported here. They weren't well represented. And we see that just in kind of a lower resolution marker along the maternal line. There's actually a lot that we can unpack here, thinking about the history of colonization of the Caribbean and the interaction with African and indigenous peoples, and how that has sort of sussed out in mitochondrial DNA. But I did do it just, again, wanted to bring up this difference, and then again connect that to community sentiments. In terms of the Y chromosome, these are the different haplogroups that we see. And again, I will draw your attention to haplogroup QM3. It shows up in this sample. You'll notice that I only have 17 people in this study. previous work that has been done with Puerto Ricans, general populous Puerto Ricans, where the sample sizes, you'll see studies with 400 people, 1,000 people, none of them show QM3. But in this small Afro-Puerto Rican community, it shows up here. Again, it's telling us that something happened in the past that we aren't aware of. There was interactions between indigenous, particularly men, and these African communities, and the genetics is really pulling that up. When we look at autosomal ancestry in these communities, again, I'm really just showing you these differences between Afro-Puerto Ricans and Puerto Ricans. So it really matters who you ask to be in the study and how you ask them. Okay? How do you self-identify? These things matter. In this table here, you can see the kind of absolute difference between these ancestral components in Afro-Puerto Ricans and then Puerto Rican sort of general populace or reference samples. And again, it really drives home the point that communities were saying with these initial studies, that they weren't really well represented initially. Okay? So there are stories to be told when we start to look at some of these subpopulations in the islands. So those are the results of the genetic aspects of the study so far. Again, this is an ongoing project. We'll be adding more data, and there's actually several analyses that I didn't share with you. With the easing of the pandemic, we actually are going to return to Puerto Rico in June and collect additional samples from the community and revise these analyses. So despite the delays in getting the project completed, study participants and other community members actually continue to be very interested and supportive of project goals. And again, it's because the project goals align with what the community wants, the community's agendas. So the images I'm showing you here were taken last October when I went with my collaborator, and we participated in a weekend-long conference at COPI. Here we shared study findings with the community and actually interacted with study participants and other community members. In this particular conference, it featured a cultural exchange between COPI's dance company. So this is actually a group of artisans, so dancers, musicians, painters, sculptors, etc. They all kind of work out of this community center. But in this conference, they had invited a Garifuna dance company. So they were known as the Chief Joseph Chatioe Dance Company. And again, it's a Garifuna community residing in New York. And here, I think I can get this to play, there's a little snippet of the culmination of the conference. And this is when the groups kind of met and danced together. Okay, so that's kind of the highlight of the event. So what you were hearing is actually bomba music, and there's actually a lot of similarities between bomba music and Garifuna music. And the dancers, actually, there's a lot of similarities and some differences between the groups. But there's this really interesting weekend where people came together and they saw these commonalities, and then I got to throw some genetics in there, so it was phenomenal weekend. So in sharing the results of the studies that I'd done previously in St. Vincent and the ongoing work in Puerto Rico, conference attendees actually reported being very emotional and moved with a deeper understanding of how their community histories were reflected in their biology, how it was inscribed in their DNA. In addition, the presentation of the genomic work gave conference participants novel perspectives on the histories of their respective communities. It provided another way for them to think about and grapple with the histories of trauma and triumph that characterize many African-descended communities in the Americas. So in considering the cases of Puerto Rico, the First Peoples community of Arima in Trinidad, and St. Vincent's Garifuna, the use of community-based participatory approaches has helped to ensure that those who have been traditionally marginalized have more autonomy about how research on their communities is conducted. By engaging with communities and research from the outset of the project, we actually sought to address questions about the origins and ancestry that were of interest to these communities, and also had sort of scholarly traction in that it allowed me to learn about how dynamics of human experience can work to shape and influence human diversity. In using this approach, the intersections between culture and biology became that much more apparent. This recognition of the intersecting nature of culture and biology, it has implications that actually go beyond anthropology. For example, such a perspective might be useful for understanding more about how the impact of environment, broadly understood, how that becomes biology. That is, how environment can get under the skin and impact health outcomes. Ultimately, by making and keeping participant community interests central to the study, researchers can work to ensure that science is used in service of people and not for science's sake itself. In particular, if communities of scientists are serious about improving diversity not only within our data sets, but also in who practices and engages with genomics, then it will be imperative that there is critical thought about the relationships between those who study and those who are studied. And with that, I will say thank you. First to my community partners, Ricardo Barat Hernandez, Zodia Brown, Marlon Stevenson, Wendell Walters, and Marty Cruz Rivera Clemente. I'm also grateful to all the people that actually make this work happen. So my field work assistants, my graduate students, my lab manager, in addition my collaborators, Doctors Gabriel Torres Colon, Dr. Maria Nieves Colon, and Dr. Melinda Aldrich. They've all been instrumental in kind of getting this work going and keeping it going. And of course, we always thank the folks who pay the bills, so College of Arts and Science. Thank you. At Vanderbilt, at Notre Dame, at Nat Geo, and the University of Pennsylvania. So with that, I'm happy to take questions and enjoy conversation. Thank you.
Really interesting talk. I'm wondering, are the samples anonymized, or can and if not, if someone is interested in knowing the particular ancestry of their family, can that information be given in this sort of study?
So, it's difficult to really think about anonymity when you have sample sizes this small.
I suppose, yeah, of course.
Yeah, and then I mean, honestly, it's the Caribbean, so everybody's in everybody's business anyway. So, I mean, we can have the illusion, like I could take off names and identifiers, but people will, if you know the community, you'll be able to kind of figure these things out. So, I do my best to kind of manage this, but the fact is it's a small community.
So how, like, does it occur that someone really, they want to know about their family, and do they get information sometimes that, disputes what their own impression was?
Yeah, I do my best.
Are there surprises?
Yeah, so sometimes people inadvertently want paternity tests, and I don't want any part of that. People, sometimes they have secrets for a good reason, and I'm not interested in uncovering that, so I do my best to kind of avoid situations like that. But yes, I mean, people do get information about their own lineages. So, some folks will be very invested in a particular identity and get the genetic results back and find out that the genetics don't really support that. So then we have to have a conversation about what that actually means. How much can we interpret from genetics? And what it actually means to identify. Is biology a precursor? Do you have to have it? Is it necessary and sufficient just to have it? Or is it more about how you were raised, how you understand the world, how you engage in the world? So it's a matter of sort of managing expectations, and in some cases really deprioritizing science as as just another way of knowing about the world.
Yeah, because in a way, then, if that happens, then, as scientists, you're having some lasting, potentially negative impact on people, and so it's obviously a complex issue, but you're influencing the lives of the people that you may introduce some disturbing information there.
Right, which is important to kind of maintain that relationship even when the study's done, right? Because the data can be queried in different ways that the community might not have been happy with those particular questions being asked, but you need to know to go ask them, hey, is this okay? And then when they say no, because eventually you'll get a no, you have to be okay with that. Even though, that means that's one less paper you write. It has impacts on your own kind of scholarly work. But who are we doing this for anyway?
That was a fantastic talk, and I was particularly struck by the Trinidadian population talked about. And being Asian myself, I was struck by the small percentage of Asian markers in that group. But you mentioned it was from probably an Indian population, although it seemed like the data also said it was purely Indian, Southeast Asians really partitioned differently from East Asians.
So
And
you have a
yeah,
great
no,
eye. I actually skipped over a lot. Trinidad is fascinating. Fascinating, yeah.
So, I mean, that would suggest that Trinidad is a melting pot, quite distinct from the rest of the other Caribbean regions.
Yeah.
My question, though, is the Asian introduction had to be at a different, much more recent time point than, say, the African proportion.
Mm-hmm.
What's the time frame for the introduction of the Asian population, and how quickly does that eventually permeate into the genetic ancestry data?
So emancipation happens in 1834 to 1838, because people want to take time, I guess, apparently to free folks, but after that, there were these labor voids, right? Because formerly enslaved people were not doing the same, refused to do the same labor that they did before. So there, and keep in mind kind of the context of the world at the time. The British Empire was everywhere, including in Trinidad and in different parts of Asia. So it's
Okay.
after emancipation that you start to see the influx of primarily South Asian laborers, but there are also East Asian laborers. So you do have an influx of Chinese immigrants coming in, working, and there are Chinese Trinidadians, which is
Interesting.
it's fascinating. You see it reflected in food and music, and it is yeah. But it's kind of another topic to explore because of there's social structures that exist that kind of keep communities segregated in some ways, in some
Yeah.
facets, but not all the time. So that's another area. I did gloss over that, but you have a keen eye, so I'm happy you saw that.
Because I also noticed you showed a slide of Belize, and I've been to Belize, and I noticed there's an enclave of Chinese Belize there too. Do you see this also in Belize, or have you started looking in Belize?
I haven't looked in Belize, but I know just sort of culturally, and again, not to over homogenize the Caribbean, but even like different Chinese foods, there's a Caribbean version because of immigrants coming in and sort of influencing and mixing things up and making kind of traditional foods in local ways.
Interesting.
Yes, there's a lot to be studied there as
Okay.
well.
Do you see mixing between island ethnic populations due to like volcanic eruptions and evacuations and
So
I know
terms of-
St. Vincent just had a big eruption last year.
They did, yeah, and actually, those communities I showed you, Oya, Fancy, and a lot of the people in that area actually were evacuated, and I sent masks to my community partners there, and clothing, etc. But yeah, a lot of people were displaced, and conventional or traditional homelands were kind of lost as a result of that eruption. Now, in terms of mixing, it's harder for me to answer that because I've never really thought about that question, but I do know, based on some of my visits to other islands, that indigenous people have been moving across these islands for a long time. Like, it's bold to me to sort of stand on the shore and say, I'm gonna go over there, I'm gonna get in a boat and I'm just gonna go. But they did. So there was a lot of movement between islands, and this continues to happen. I was talking to people in Dominica, I think it was, and they were talking about like, their grandfather and their great grandfather would just build boats and they would go. And it didn't matter that, that this island was French and people would move. So I would imagine things like volcanoes, major geologic disruptions would cause people to move around because that's what people have always done.
Yeah, so I'm thinking about this school of thought that's coming to bear in the genetics community, that people should stop referring to ancestry in continental levels and be a little bit more precise about it, because otherwise has the potential to reify this idea that race is a biological construct, which it of course is not. And I'm wondering how that school of thought overlays with the type of work you do in partnering with communities. And I noticed that you mentioned talking about race versus genetics with your community partners, and I was wondering if you could give us a sense of what those conversations are like and how this emerging school of thought influences it.
So this is a situation where you meet people where they are. So there's already these ideas about race and identity and continental ancestry that exist. So you have to be very careful, right, as to how you approach this, but you want to talk in terms that people understand. Okay, so as I do this, I am very careful to talk about, okay, you have ancestors who may have identified it this way, and that may or may not have bearing on how you self-identify. So I'm very careful about how I speak about it. In addition to, I don't want to oversell the technology. A lot of the communities I work with, they actually want to know where in Africa their ancestors came from. And it's a much more complex answer than we have the technology to really piece apart, and we may not be able to because people have been mixing for so long. And then ethnic labels, identity labels, those things change within generations. And they don't necessarily have genetic markers that attach to it. So explaining those concepts can be complicated, but it is part of the conversation. To do it in ways that are respectful, that you're not talking down to people, but again, sort of meeting them where they are. In terms of thinking about these connections between self-identification, these social ideas of race, and medical genomics, personal genomics, these are all conversations that have to be carefully had. And we need to understand or think more critically about when it is time to start talking about race and when it is time to be talking about genetic ancestry, and when that matters.
I have one more quick question. What happened in Haiti and Jamaica, where there's very little left of indigenous American ancestry markers?
So, I have, these are completely unsubstantiated thoughts, but I'll
Okay.
share them anyway.
Okay.
I think with some of those studies, it's really a matter of who is sampled. I think if you, like in Jamaica, because I've actually done some work there are contingents of people who identify as Taino. Okay, so I think if you sample them, you'll probably find higher amounts of indigenous ancestry. I didn't talk about it, but I did some work with the Accompong Town Maroons. So their Maroons are descendants of enslaved Africans. We can debate whether they were enslaved or not, but they were Africans who refused to be enslaved and instead ran away and set up their own sort of semi-autonomous communities, waged war with the British, won, and then had sort of these semi-autonomous communities. Historically, there's information that suggests that the very first Maroons were actually indigenous people moving away from the Spaniards, right, in their abuses. And then later, African descendants joined them. And it kind of makes sense. You're in a new land, new flora, new fauna. How do you figure out how to live here? You just ask the folks that have been here for forever, okay? And they formed communities. So the work I did with the Accompong Town Maroons, they have oral histories saying that the first Maroons were actually Taino, that they were indigenous people. So it was really interesting to see if we could detect any of that in their DNA. Again, really small sample, one community, and I did see it along the maternal lines. I do suspect that if I were to return and include other Maroon communities that are in other parts of the island, particularly those in the Blue Mountains, we would find more indigenous ancestry. So I don't think it's completely gone. I think those islands were definitely impacted. The ways in which the French and the Spanish practiced colonization, they would basically clear the island first. So either kill folks or export them to other islands as slave labor, and then bring in enslaved Africans. Regardless, there are pockets on the island who likely have genetic ties and then self-identify culturally as indigenous. So it might just be a matter of sampling. I wouldn't go out and say that they're not there.
Okay.
So when you're engaging with communities, what has worked well at helping people to understand the complexity and the uncertainty and the implications of the data that you're doing within the broader context that you're trying to work? Is there any kind of specific or general advice that could help other people doing these sorts of?
Yeah, time. Taking the time to build relationships with people, and then doing your best to kind of understand how they see the world, and then using your knowledge of genetics, putting it in terms that make sense in their context. Okay, in order to do that, you have to be able to hang out with people. You've got to be able to kind of interact and see how it is they make sense of the world, and then you too can make sense of the world. This example of finding genetic ancestry that doesn't necessarily align with a person's ideas about themselves, okay? Knowing how important those connections to the community are, you can't just put the genetic data on their lap and then walk away. You do have to talk about, well, there's different ways of knowing, and these are the limitations of genetic data. It's not the end-all be-all. I mean, this is just, what this shows. and then couching it in those terms. But again, the important part is really knowing what the community wants, and then how your work fits into that, and not the other way around.
Let's thank Dr. Torres for a fantastic talk.